Hello my gentle and of course very modern apes, welcome to Gutsickgibbon. This is my YouTube channel where I talk about the latest discoveries in topics that I find interesting with relation to science. Usually it's biological anthropology, primatology, evolutionary biology, things of that nature, and I also spend a whole lot of time debunking pseudoscience. And once upon a time, I spent a whole lot of time debating and sometimes reviewing debates. If you YouTube search gutsickgibbon debate, you're going to find a lot of results, and that's because I had a very long period of time, my debate bro arc, where I debated every creationist on the internet that was willing to debate me.
All due respect, you got no fucking idea what it's like to be number one.
I don't do debates anymore, but that's only because I debated every creationist who was willing to debate me, and I'm only being a little bit hyperbolic on that. There are a couple that I never really got to have, but that's mostly because I had issues getting them scheduled, finding a platform that I was okay being on, and just having the time to really get into it. Many of you know this, but I'm a fourth-year PhD candidate, so this channel has mostly operated while I was getting my master's degree and then my doctorate, so time comes and goes. But suffice it to say, if I have the available time and there's a creationist that wants to debate me, especially if they're a professional, I'm gonna drop everything and try to make it happen. And that has happened. Here in a few weeks, I'm going to be debating or discussing with prolific creationist advocate Jerry Bergman. Bergman is interesting to me for a couple of reasons. One, because he's obviously prolific. He's done a lot of writing on the evolution creationism controversy, and he's associated with many of the usual suspects, like the Institute for Creation Research or Creation Ministries International. But what I find most interesting are the subjects that he actually writes about, the topics that he chooses to cover. In addition to the usual things that creationists like to talk about, like irreducible complexity, and I'm not talking about the Behe style, I'm talking about, like, Bergman will talk about how carbon atoms are irreducibly complex, it's a long story, and vestigial organs, he has spent a lot of time slandering the name of Charles Darwin, calling him, like, a sadistic Machiavellian freak, and also a huge racist, and Bergman is perhaps best known for his long-standing claim that creationists are being systematically discriminated against in academia. I think that it's fair to say that this crusade stems from the fact that Jerry Bergman was denied tenure at his home university of Bowling Green State University, and he claims that the reason for this was that it was because he was a creationist. He said they denied him tenure because he is a creationist and it is thus religious discrimination. He took the university to court over this and did not win the case because, according to court documents, he was actually denied tenure because he did not have the credentials that he said he had. This has not stopped Bergman from writing many books on religious discrimination, specifically with regard to young earth creationism at academic institutions across the United States. My idol, the late Ronald Numbers, in his incredible expanded edition of The Creationists, covers Jerry Bergman and describes the situation like this. Bergman's compendium of alleged persecutions often lacks the documentation needed to support his claims, and in a number of celebrated cases, additional testimony either falsifies or substantially modifies his account.
Of all the things in the world that have never happened, that has never happened the most. That is the most didn't happen shit of all time. What a bunch of baloney. Actual horseradish.
Now, it's my opinion that Jerry Bergman was not fired because he was a young earth creationist and that there is not widespread discrimination against young earth creationists across the United States. I think the fact that so many creationists just work at universities and have jobs and talk about how they have jobs at real universities, like sometimes in an attempt to legitimize creationism, is pretty excellent support for that. The truth of the matter is most universities don't really care at all if you're a creationist.
I'm trying to stay out of the debate. Really? I mean, you know, best of luck.
Good point.
Why not? I mean, other than being wrong. Why not? You don't have to be right about things. So bourgeois.
But whether there is or isn't, and whether Bergman has honestly or dishonestly portrayed the situation in his various books, is mostly irrelevant to the topic of evolution versus creationism and his arguments against evolutionary theory or for creationism. Dr. Bergman has some impressive credentials, so I'm interested. What are his arguments against evolutionary theory? Why does he believe that young Earth creationism is a more accurate model for the state of the natural world? So I want to go through some of Bergman's debates and videos. I want to assess his accuracy when dealing with a lot of concepts regarding evolutionary theory and creationism. And this kind of serves two purposes. One, it gives you the background that you need for Jerry Bergman and his arguments prior to hopefully consuming my debate with him here in a few weeks. And two, it gives me an opportunity to investigate my interlocutor. One thing you gotta know about me is I like to research, I like to dig, I like to get my simian little fingers all over someone's background so that I can know what I'm about to get into. And I do that, I think, quite well and quite thoroughly. So 3, this can kind of act as a little cheat sheet for you, Jerry, for our upcoming discussion. You can use this to know where I'm going to probe. I mean, I hope Jerry watches this. I hope he uses it as a cheat sheet. My mom actually told me that she worries that creationists might not actually watch my videos because of the Demon Days poster that I have in the backgrounds of some videos, because it's this demon. That's just an album from a band called Gorillaz. It doesn't have anything to do with actual demons. I certainly hope creationists are reasonable enough to know that if you listen to music that has a scary word in it, that doesn't mean you're like evil or trying to subliminally convince an audience to believe evil things, especially because if you're conspiracy brained enough, there's a lot of stuff in this room that you could use to erroneously come to the conclusion that I'm like a lizard person working for the deep state. What I would like to do is just go through some of Jerry's appearances online, some of his presentations or debates, and kind of just assess them, go through them, dissect them, if you will. And I think that will be useful for all parties involved for this upcoming discussion. So, grab a snack and a drink, get cozy. I'm not known for my brevity. And what the heck, let's debunk some creationism. So Jerry Bergman isn't dissimilar to Duane Gish in the sense that he has had a lot of debates. One of the oldest videos that I can find of a debate that Bergman has had was with P.Z. Myers, an American biologist and a pretty prominent member of the sort of evolution side of the creation-evolution controversy, which he's been involved with for many, many decades. And it was obviously a debate from like 2009, which was quite some time ago, although P.Z. uploaded it to his YouTube channel much more recently. And the introduction that he provides to that debate, sort of retrospecting on that debate, does not bode well.
So in 2009, I did a debate with a creationist by the name of Jerry Bergman. And honestly, it was terrible. It was just one of those experiences where I just realized that debates were a total waste of time.
Uh-oh. Ugh, that's not good. The problem, PZ goes on to point out, is that he did all this legwork, all this research on court cases and definitions because the debate was on the nature of intelligent design. Should intelligent design be taught in schools? And that makes sense because this was in 2009, which was pretty close to the famous 2005 Kitzmiller v. Dover trial. And PZ laments that he put in all this work for researching and reading up and boning up, if you will, on all of these different topics that should be relevant to that topic, and
it was all wasted time because the guy's a loon. He just went off in all directions.
This is true. And when you watch the debate, as I have, you will see that Jerry doesn't really stick to the topic of should ID be taught in schools, and instead he spends a whole lot of time kind of gish-galloping, running all over the place, and talking about all of these different, perceived problems that he feels exist with evolution. And one of those topics is the complexity of the carbon atom.
In fact, there was a teacher in Ohio who was terminated, and in the court papers, a number of his teachers said he was let go because he, quote, taught the periodic table. Because the periodic table is irreducibly complex. A good example, to get carbon-12, you must have what? 6 protons, 6 neutrons, and 6 electrons. If you have anything else, you will not have a carbon-12 atom. It is clearly, unequivocally, irreducibly complex.
What? Irreducible complexity is an argument used within intelligent design, this proposed alternative to evolutionary theory, that some things require a creator and could not have evolved. The idea is that some structures or some pathways are irreducibly complex. They can't have evolved because each part of the system or of the structure is necessary for the structure to actually perform its function. The classic example is a mousetrap. A mousetrap is composed of 5 parts, and if you take away any of those 5 parts, the mousetrap can't function to catch mice. The problem is, that's not how evolution works. A structure or a pathway does not necessarily, and in fact almost always doesn't, perform the same function throughout the entirety of its evolutionary history. Feathers didn't evolve for flight. They started off as thermoregulatory or display structures, and then they were co-opted for flight later. We know this because the first feathers show up on dinosaurs that cannot use them to fly. And this is the case for every single example of irreducible complexity that has been put forward. There are also examples of systems which match the definition for being irreducibly complex that exists today, which we know did come about by natural means. The example that I always give is that of an ecosystem. An ecosystem requires every single population of given species within it to engage with one another in specific ways, and if any of those species goes completely extinct overnight, the ecosystem collapses, or at least drastically changes and no longer performs in the way that it did before. And yet, we have watched ecosystems form on areas where ecosystems weren't previously, like primary succession on an island, on a volcanic island, as it becomes populated by organisms for the first time. So irreducible complexity doesn't work as an argument for intelligent design because each subsequent step doesn't have to perform the same task on an evolutionary history, and we have seen irreducibly complex structures or systems be broken down into various parts, each of which is functional in its own right. But what Dr. Bergman is talking about here isn't irreducible complexity, at least not in the sense that any intelligent design proponent has ever meant it. And the reason is simple. Carbon atoms aren't alive, and they don't evolve. Like, carbon forms all the time. We observe this happening. So this isn't an example of irreducible complexity. If anything, it's an example of fine-tuning, I guess. But PZ picks up on this, and he's kind of like, what are you talking about? And when they have a sort of cross-examination, he poses the question directly to Dr. Bergman.
Well, I would like some explanation. How can you say that the carbon atom is irreducible complex? What is your personal definition of irreducible complexity, and how does it differ from Mike Behe's?
Okay, I correspond with Mike Behe on this, and so we agree.
Bergman goes on trying to justify this for a little bit, and he finishes his response with something that doesn't make any sense at all to me.
And the only way one would not be irreducibly complex is if you can go down to the point where you have a fundamental particle, like a quark or a lepton, and they would be irreducibly complex. But everything above that would be irreducibly complex, including life, watches, radios, etc.
To which PZ responds with what I mentioned earlier, that this is not irreducible complexity. It can't serve the purpose that Behe initially dreamt up for it.
But this is defeating the entire intent of the irreducible complexity argument. Because the irreducible complexity argument is being used to argue that structures couldn't have evolved in particular ways. But what we discover is that things like carbon atoms could have arisen and did arise by natural mechanisms.
Right, so the carbon example doesn't work because it doesn't meet the criteria for irreducible complexity as it is applied to biological systems, which it's basically only ever been applied to. And then also it doesn't work on top of that because we have watched carbon atoms form. Like, by this definition, a water molecule is also irreducibly complex, because if you take away one of the hydrogen atoms, it's not a water molecule anymore. And yet we see water molecules form all the time. But this is why I think Bergman doesn't really understand irreducible complexity, at least as it is used to bolster intelligent design, right? Because he goes on to give these two steps that something must make in order to be considered irreducibly complex. And the first step is that it must be irreducibly complex. And the second step is that it can't have formed naturally.
We have two steps. One step is something irreducibly complex. The next step is, could it come about by natural means? And of course, relative to the elements, of course, it can come about by natural means. But that doesn't mean that it's not irreducibly complex.
I think Bergman thinks that irreducible complexity is just any definition of a thing. It's any state of a thing, where if you change something, it isn't defined as that thing anymore. How does that help intelligent design? And PZ points out this exact fact.
Okay, it doesn't appear that we're going to reach agreement.
No, actually, I'll agree entirely with him. It just means that irreducible complexity is no longer a useful argument for evolution.
Right.
That's fine with me.
Okay. Yeah, my understanding. Okay, I'm not going to.
I love the moderator. Who is he? I hope he's doing well. But Bergman's entire perspective on all of this is so unbelievably bizarre, because he goes on to say that no, actually, it is a good argument for intelligent design. You just have to go step by step.
Can I answer that?
Yeah, sure. Quickly.
I think it's an excellent argument for intelligent design. A very good argument, but you have to go a step at a time, and I can't do it in 30 seconds.
Okay.
But it's like, we've observed the formation of carbon, like, step by step, so how exactly does this work? It seems like he's got one foot in the biology realm and one foot in the fine-tuning chemistry realm here. To the best of my understanding, Bergman sort of lumps in fine-tuning with irreducible complexity, which is not a common way of using that phrase. But he consistently, and we will see this again later, appeals to this notion that unless you can explain absolutely where everything in the entire universe came from step by step through naturalistic means, then it's irreducibly complex, therefore God.
But that doesn't mean that it's not irreducibly complex. That simply, now we're talking about how things came into existence, which is another question.
In fact, this, God of the Gaps, is going to be a reigning theme with Bergman moving forward. It's going to be God of the Gaps, Darwin bad, and a persecution complex. And part of why I wanted to start so early, so long ago, in the misty dead eons of time in 2009 with this debate, is because we're going to see some of these themes pop up in these more modern debates as well. And before we leave PZ and Bergman here, I do want to show you this one part that just tickled me pink, where Bergman asks PZ if PZ knows of any out of the closet, and that's how he frames it, ID proponents who have tenure. And this is how PZ responds.
You know, any out-of-the-closet ID supporter or creationist who was openly out of the closet and who got tenure at a university?
No. And the problem there is?
So you're saying they should be terminated?
I'm saying in order to get tenure, what you have to have is basic competence in your discipline. You have to have a publication record in your discipline. You have to be able to interact and work with your colleagues in your discipline. And somebody who rejects the basic principles of biology is not somebody we want teaching biology. So yeah, I think it's entirely appropriate that somebody who promotes a theory that is not a theory, that has no evidence, should not be given tenure in a university department.
Oh man, ouch. That hurt me. Did you hear the audience when PZ said you need to have basic confidence? They oofed. You can hear them oof on camera.
In order to get tenure, what you have to have is basic competence in your discipline. You have to have a publication record in your discipline.
Eesh, bow, wow. That hurts me. To be fair, though, I mean, you know, PZ has a pretty good point. When I was talking earlier about how I don't think creationists are getting, like, fired or denied tenure for being creationists, I sort of meant, like, at universities in general. But it probably is the case, and probably should be the case, that if you are a young Earth creationist, you should not get tenure if you are working in a biology department, or perhaps a geology department, an astronomy department, an anthropology department, a chemistry department, a physics department. Really, if you're a young Earth creationist, you probably shouldn't get tenure, sorry. I guess if you are a young Earth creationist in any of those departments, and you can competently teach evolution to your students, which is a necessary concept to know and understand to pass any biology course, like down to the high school level, and you're publishing in conventional journals and you're publishing good work in conventional journals, I guess if you're doing all of those things and you're like having a good rapport with your students and all of that is true, then maybe you can get tenure if you are a young Earth creationist. I think the problem is it's very unlikely that you are going to both be a young Earth creationist and also do those other things. I think that's what PZ is getting at here. So now that we've got our fill of an older debate posted to YouTube in 2009, but recorded much, much earlier, again, back in the misty annals of history, let's move more recently. I want to talk about a debate that Jerry Bergman had with Dr. Chris Thompson within the past 6 months. We're not going to do the entire debate because we have more to get to, but I'd like to just highlight a couple of pieces here. Our debater in the affirmative for evolutionary theory being responsible for the origin of species is, as I said, Dr. Chris Thompson, and I love this guy.
Hi, my name is Chris Thompson, and I'm a professor of neuroscience.
Dr. Thompson re-uploaded the debate, which was hosted on the Standing for Truth channel, over on his own channel, and he gives a little bit of an intro about how he feels the debate went. He talks about what the topic is. He talks a little bit about Jerry Bergman and what he's best known for.
One of those big things is gathering evidence of creationists being discriminated against in academia and other places. The debate that we had was on the origin of species. This occurred over at Standing for Truth's channel. I think the debate went very well. I think that it was well moderated.
Ooh, I concur, Chris. I also think the debate went really well. We're going to talk about some of that here in a minute. And I know it's kind of unpopular to give Donnie compliments, but I also think Donnie is pretty good at moderating. So good for you, Donnie. Mr. Badinsky's talents aside, Chris then talks about how he feels the debate went for him. And he says something that when I first listened to this video, when he said this, I was like, oh man, either this guy is like really overconfident or he absolutely slated in this thing. Because the thing about doing debates is that when you finish them, you tend to look back and think like, oh, I could have done this better, I could have done that better, I could have said this, I could have said that. On the rare occasion that you're like, man, I absolutely killed it. At least for me, I'm not thinking that unless I actually did. So when Chris says this, I got excited.
I also think that I, essentially mopped the floor with him.
This got me excited because I don't know Chris Thompson. I don't know his debating skills. I don't know how much he knows about young Earth creationism, because a lot of people don't know this, but to successfully debate a young Earth creationist, you have to actually know what they argue. It's not enough to just be well-versed in the conventional science. You have to actually be familiar with the young Earth creationist models, with their hypotheses, with what they propose, the various idiosyncrasies of each specific type of young Earth creationist. You have to establish what the specific young Earth creationist you're debating actually believes, so you're not strawmanning them, all that sort of stuff. So when Chris said this, I was like, okay, either this guy has really got something special for us, or he's overconfident and delusional and isn't remembering the debate properly. And I had a really good feeling that it was probably the former.
If you like debates where the creationist ends up squirming most of the time and trying to backpedal on just about everything, end up telling me that I have a bunch of good points, that's this debate for you. So it's about 3 hours of that.
3 hours? Child's play. So let's get into this a little bit. I want to get into it because I'm a fan of Chris Thompson. I'm a fan of Dr. Thompson's work here. I think he's done a really bang-up job, and I actually learned quite a bit from this debate myself. So let's talk a little bit about some of the highlights before we move on to our next Jerry content iceberg, our next Jerry Bergman iceberg. There's definitely something there. Our next icebergman. There it is. So like I said, Jerry Bergman, Dr. Jerry Bergman, is a young Earth creationist, and so in this debate that he's having with Dr. Chris Thompson, they are arguing about the origin of species, right? This is basic evolutionary concepts. Can we explain the origin of species using evolutionary theory? And taken back in time, is common descent supported as well? So Dr. Thompson gives his opener, and I think it's really good. Chris has to spend some of his opener explaining what evolutionary theory is in case anybody in the audience doesn't know. And I think that's good practice, but it also puts him at a disadvantage because you're having to explain something instead of getting to the evidence that actually supports it. Right, so evolutionary theory is a theory that explains why organisms are diverse today, and taken back in time, it explains where organisms come from. They converge on a last universal common ancestor in the same way that you and your siblings converge on a last common ancestor in the form of your parents, and then all of you converge on a last common ancestor in the form of your grandparents. It's just mathematics. And then he goes into how evolution works. What are the mechanisms for evolution? How do we actually get changes in allele frequency in populations over time? And he talks about how new mutations are responsible for adding varieties to populations. And then varieties within populations, naturally, some of those individuals, some of those varieties are better at reproducing than some others within their populations. That creates what's called differential reproductive success. The ones that are better at reproducing do leave behind more offspring, and those offspring typically have the genes that gave their parent an edge in the first place. And over time, if the pressure stays the same in that environment for those specific traits, and what is giving organisms an edge, what's making them more fit, is context specific. In some environments, it's going to be some characteristics, and in others, it's going to be others. What's beneficial in a desert isn't necessarily beneficial in the Arctic. Over time, populations will change in response to environmental pressures. There are also non-selective mechanisms at play as well. Typically, you talk about things like genetic drift, which is the random fluctuation of alleles due to chance events usually. And then we also have gene flow, which is migration. How do populations exchange alleles with one another, and what happens when they don't anymore? It's all really boilerplate stuff.
I don't want to say natural selection is the only mechanism of evolutionary change. There's also genetic drift, migration, also mutation. Mutation is an important thing. I know Jerry's going to be talking about that.
Yes, he is. And Jerry talks a whole lot in a lot of these intros and debates and in a lot of his presentations about mutations and about how when he worked with cancer cells and when he was in cancer research, mutations are just so bad all the time that it's absurd to him that anybody could think that mutations can effectively be beneficial at all and eventually lead to things like speciation and the origination of like novel characteristics or novel body plans. So it's important that Chris is knocking this out right off the bat. And he talks about how these beneficial mutations work, and as somebody who works in a lab and who is, in neuroscience, I think that his testimony here is very apt.
There are mutations that can be beneficial. Most mutations are neutral. They really have no effect. Of course, then some of them can be deleterious and cause cancer, just as Jerry already stated, and I'm sure he will be talking about. But there are beneficial mutations. We can see these examples from artificial selection experiments. So you can go into the lab and try to develop molecular tools, not through like designing features or like trying to create specific mutations, but instead just inducing a bunch of different mutations in the construct that you're trying to mess with, and then look to see which one actually confers the benefit for what kind of tool you're trying to develop. Then you take that and then do another round of mutation and another round of mutation and selection, mutation, selection, mutation, selection. Through that process, you can create whole new genetic tools. This kind of technique is used all the time in the lab.
So that is insanely cool to me, and I did not know that. I had no idea that mutation and natural selection played such an enormous role in biomedicine in this way. I knew, obviously, that understanding it plays a massive role in understanding how diseases change over time with antibiotic resistance and how we can best combat that through drug trials and things of that nature. But I didn't know that we actually used it in medical trials to create genetic tools. I guess I was under the impression that I think many people are, which is that people get in there and edit genes manually. But nope, they use good old-fashioned mutation. So they trigger mutagenesis to get all of these varieties in the first place, and then they just let natural selection, or really artificial selection, play out. That's so cool. And I know what young earth creationists will say already. I know what the haters will say. They'll be like, well, it's artificial selection, so that doesn't count. But no, it does count, because the mutagenesis isn't controlled. They're just trying to trigger a bunch of random, or rather probabilistic mutations. They don't know the specific mutations that are going to happen. Then they artificially select the characteristics that are beneficial to, the cell or whatever in that given moment. But artificial selection is just natural selection imposed by an agent. So why wouldn't this kind of thing happen in the wild? If these types of mutations happened, out in some little warm pond somewhere, and they were beneficial. And remember, what's beneficial is context specific. What is beneficial in the lab is not necessarily going to be beneficial in the wild, but it might be, which means if that mutation does show up and confers a benefit within that context, those organisms would survive better than their fellows. You don't need a human there to kill off all of the others. Differential reproductive success will take place, and then you got a stew going. Chris picks a great pair of organisms to point to say that, yeah, we see this in the wild as well. He picks a bush dog and a maned wolf, which is a pair of organisms that creationists, young Earth creationists, would believe actually do share a common ancestor in the form of a created kind roughly 6,000 years ago. More on that in a moment.
We can also see evidence of beneficial mutations that have occurred in the recent past by comparing closely related species that both young Earth creationists and evolution proponents would agree are actually related to each other, such as the maned wolf and the bush dog.
Most people who watch this channel know this, but on the off chance that you don't, young Earth creationists don't typically believe that every single species was separately created by God 6,000 years ago in the Garden of Eden. Instead, they believe that 6,000 years ago in the Garden of Eden, God created archetypal kinds, roughly at the family level, which then descended into their modern counterparts after a bottleneck when Noah's flood happened 4,400 years ago. So God didn't create, every cat species. He created an archetypal cat that then rapidly evolved into all modern cats. And that includes things like lions and tigers and saber-toothed cats and leopards and sand cats and desert cats and all sorts of stuff like that. Which is why Thompson's example here works so well, because bush dogs and maned wolves are morphologically extremely different, and yet they're one another's closest living relatives. And so creationists have to allow them to share a common ancestor. They're both within the dog kind. And so, under that lens, even Jerry would have to accept that a whole lot of beneficial mutations can happen to get a bush dog and a maned wolf from some kind of generic ancestral dog. And on a rush order of just 6,000 or even 4,400 years at that.
So they also believe in phylogenetic trees, right? So within the rodent kind, there would be this phylogenetic tree as well. So this is the thing, like we actually agree on a lot. And that's what I hope that we can get into today, because I want to talk about what is the species mechanism that occurred within the last 4,500 years from the young Earth creationist model.
I think this is a great strategy because what Chris is basically saying is that conventional science and young Earth creationists agree that phylogenetic trees can tell you who is more closely related to who. It's just that creationists think it stops at some point, and conventional science doesn't. And that's sort of what Chris is going to drill down to. Where is the line drawn, and why is it drawn there? And he's going to focus on...
I want to talk about rodents. So rodents, this is the rodent phylogenetic tree. I realize that this is what evolution proponents believe, that creationists don't believe that all these animals are related to each other. But within the rodents, that we have things like beavers, squirrels, chipmunks, but also within the myomorphs, which is this branch here, we've got the mouse-like rodents, which is a quarter of all mammalian species.
That's actually crazy. I knew rodents really had sort of a handle on the game as far as biodiversity went, but I didn't know that it was a specific group of rodents, like the mouse-like rodents, which were evidently just dominating.
This includes mice, rats, gerbils, hamsters, lemmings, and voles. So let's zoom in a little bit. Okay, so we're zooming in on that box. And within the myomorphs, we have the superfamily called the Meroidia. This includes the mice, rats, voles, and hamsters. It doesn't include the birch mice or the jumping mice or the jerboas. They're found on all continents except Antarctica, and there are 6 families of muroids. And the reason why I point out the 6 families is because generally young Earth creationists believe that the families are where we have the different kinds.
This is why I love Chris's intro, because he's asking a very simple question to Jerry here. If the line is at the family level, are deer mice related to regular mice or not? Because if they're not, that sounds pretty dumb.
So this is a phylogenetic tree of the muroids, okay? So these 6 different families right here, right, are the different muroids, okay? So generally creationists would believe that these are 6 separate kinds, but within evolution proponents, we believe that they are related to each other. It comes from this excellent paper where they looked at 900 different species of muroids and created a phylogenetic tree based on their genetic similarity.
Right, so the long and short of it is Dr. Thompson is asking is Paromyscus leucopus the same kind as Mus musculus? Because they belong to different families, and most creationists think that the family level is the kind level. So they would propose that these two little guys are not related at all. A simple question, and I can't wait to get to Jerry's response, but first Jerry has to do his intro, so let's see if there's anything of note in there.
And so that's really what I'm concerned about, molecules to man. I'm not so concerned about the variation in animals as brought out quite well.
But the title of the debate is on the origin of species, right? So he's kind of just skirting the entire topic to go to something that he feels is more defensible, which is that no one has ever demonstrated in a lab approximately 3.8 billion years of evolution from simple cells to all of modern biodiversity in real time, because that would take 3.8 billion years.
There's an enormous amount of variation, and as I do more research in biology, I realize, well, horses, there are some horses about the size of a small dog, others are about the size of a dinosaur.
Which horse? And which dinosaur, right? Jerry is perfectly happy just looking at all of modern biodiversity and looking at their nested hierarchical patterns that they create genetically and morphologically that just so happens to map exactly what is predicted by evolutionary theory and extends back in time and maps across the fossil record as well. And he kind of just puts his hands up and goes, I don't really need to worry about that. His entire talk is about improbabilities with regard to mutations, right? He points to mutations and he's like, mutations are more likely to occur in some regions than in others. Lots of mutations are deleterious, and so how on earth are you going to get massive amounts of beneficial change within organisms using just mutations and natural selection? Which the question that I'm sure you're immediately wondering, and the one that I immediately wondered is, but you accept that change. Again, these guys accept specific phylogenetic trees. A bush dog and a maned wolf descend from a common ancestor under the young earth creationist paradigm. So necessarily, they accept a whole lot of evolutionary change, and a whole lot of that evolutionary change is, in fact, beneficial. So how does this square exactly? Does Jerry think that all of that change was manually instituted by the hand of God directly? Kind of. He brings up artificial selection, and he's like, but the traits that are artificially selected for, like, double-muscled cattle are not beneficial to the cattle, they're beneficial to us, which is why we select it. And it's like, yes, but artificial selection is just natural selection instituted by an agent. So if double-muscling as a mutation showed up in a natural population, and it did benefit cattle, it would be selected for, and if it didn't, then it wouldn't be. And he talks about all of these other deleterious mutations, like when animals accidentally have two heads, and he shows like pictures of two-headed turtles, and he's like, that's not necessarily good. And he talks about people with polydactyly, and he's like, that's not necessarily good. And his entire point is, and I'm sort of being hyperbolic here, but it's like, because we're not seeing incredibly beneficial mutations that like game change an entire species every single day, how are we to expect that it happened over 3.8 billion years? And the answer is twofold. One, 3.8 billion years is a lot more time than the time period that humans have spent studying this kind of stuff. And two, and way more importantly, the types of mutations that occur are not game-changing, right? They're adaptations on pre-existing structures which change slowly over time, at least generally speaking. And this is, again, something that Jerry accepts. If he thinks that all dogs are related, then necessarily maned wolves and bush dogs share a common ancestor. And necessarily there were beneficial mutations that gave maned wolves their very different body proportions than what we see in bush dogs. And the reason is because maned wolves live in habitats where those long limbs are helpful to them, and bush dogs live in habitats where those short, stumpy legs are beneficial to them. I think Jerry spent too long studying exclusively deleterious mutations, and so now he thinks that that's like the only type of mutation that ever occurs. And I also think that he doesn't understand that what is beneficial is context specific, and I also think that he doesn't understand that artificial selection is a type of natural selection. I think there's a lot of things that he doesn't understand about mutations outside of the context of cancer specifically.
Spent much of my career studying mutations.
Jerry, that's time. That's time.
And we're done.
In the rebuttal section, Chris reiterates that beneficial mutations are not like super duper rare because, again, we use mutation and natural selection in the lab in biomedicine to create designer drugs. And it's a really, really cool section. So you should please go to Chris's channel and hopefully subscribe and watch that section if you're interested in it, because it's awesome. But I want to get to what he talks about next, because he goes back to the phylogeny stuff. And this time he sort of pivots to talking about maned wolves and bush dogs.
Now, I also want to get to, I talked about carnivores in my last debate. I want to get back to that because I do want to talk about the maned wolf and the bush dog. Because we do need to talk about species, right? We're talking about on the origin of species. And I'd like to know if Jerry thinks that like the dogs are considered a single kind or not, or are there multiple kinds within the dogs? The reason why this is important is because we need to know, like, how did speciation occur within kinds? I think that's really critical to what we're discussing here. And I didn't hear anything that he had used to address that. He just talked about mutation.
It bears repeating, right? The topic of the debate is on the origin of species, and Jerry gave an intro that is not about the origin of species. It was about molecules to man evolution and mutations being deleterious. So we're seeing a repeat of what we saw with P.Z. Meyer's debate many, many years earlier, where Jerry is kind of having the debate that he wants to have, and not necessarily the debate that he agreed to have. So Chris gets back to talking about the maned wolf and the bush dog, these South American members of the family Canidae. They're one another's closest living relatives, which I actually didn't know until this debate, and they split like roughly 3 million years ago, so they're more closely related to one another than humans are to chimpanzees, and that means they're also more closely related to one another than lions are to tigers, African elephants are to Asian elephants, and of course rats are to mice, as Chris alluded to earlier. And yet, they are so physically different, and Chris does a great job outlining some of those differences.
And the thing that's crazy about that is that these two animals are more similar to each other than they are to any other canid on the face of the planet, yet they look incredibly different from each other. They're more closely related to each other than any other canid, but this is what they look like. One is short and stubby and has webbed feet and lives in the marshlands, and the other lives in grasslands and eats wolf apples. Okay. So based on the genetic evidence, it indicates that they had a common ancestor around 3 million years ago. This is the bush dog. Again, I said it has webbed feet. And here's the maned wolf. And this is the apple that it eats, right? So they have very different diets. They have very different environments they live in, but they are more genetically similar to each other than they are to any other animal.
So, just to really hammer this home, bush dogs and maned wolves are one another's closest living relatives. They are more similar to one another than many, many pairs of animals that young Earth creationists insist are related, like lions and tigers, rats and mice, or African and Asian elephants, and yet they are so physically different from one another. And what that means is that if creationists accept they're related and they share a common ancestor, those differences accumulated in real time through natural evolutionary mechanisms, and they did so in just 4,400 years. That's what we're talking about here, and that is something that Jerry has to answer for, and he has to answer as well why maned wolves and bush dogs are related, and how we can tell that, and why other pairs of animals that creationists reject as related are not, like for instance humans and chimpanzees. And they're not going to have that conversation in this debate, but perhaps it will happen soon. But again, the critical point here isn't just that these two guys are one another's closest living relatives and yet they look very different, which has bearing on all of the created kinds, but it's also that the reason they look so different is because they accumulated beneficial mutations since their common ancestor got off the ark, according to the young Earth creationist model, which necessarily means that young Earth creationists don't just have to entertain the idea of beneficial mutations, they require them for their model, something that Jerry has just outright denied the prevalence of in his entire opener.
And so in this study, they fully sequenced all the genomes of these animals, and they were able to identify what are called private alleles. And they found more than 100 different private alleles in bush dogs and maned wolves. And a significant portion of them, nearly 10% of them, are related to just things like limb length. So one thing that seems pretty clear is that if they are shared a common ancestor, then because they have private alleles, these have to be new mutations. They cannot be mutations that were found in the original pair that came off of the ark. It's impossible. These are new mutations. They have to be new mutations. Okay, I'm going to say that again. These are new mutations. They were not on the original ark.
Chris talks a lot about the types of unique mutations that exist in Maine wolves as compared to bush dogs, and he focuses on differences in limb length, which I think is pretty intuitive when you see what these two critters look like. But Maine wolves also have experienced changes in their metabolism so that they can better digest the fruit fibers of the fruits that they eat, which other canids obviously don't have because most canids aren't frugivores. That's so cool, and I know you're wondering, I really don't understand how Jerry can respond to this. Like, that's just so clearly a beneficial mutation.
So, Jerry, you have to be able to explain this. How do private alleles come about in these canids when they obviously must have shared a common ancestor?
Jerry's answer is just that he's okay with all of that because you need a system for organisms to adapt.
Okay, I have no problem with these minor changes, these minor, there has to be a system which allows animals to adapt because we have so many animals and so much adaptation is necessary.
Boy, I wonder what that system could be. You mean maybe like evolutionary mechanisms? What happens if you take those evolutionary mechanisms that have been allowing for speciation and adaptation forward, and then you take it backward in time? What happens then?
What I'm saying is, how does a neurotransmitter evolve from nothing? And the area I've looked at it, for example, is the evolution of organs in humans.
Okay, so the goalposts have now shifted. Thompson gets a point, and it's no longer that mutations are almost always negative and beneficial mutations are very rare. It becomes, okay, well, there does need to be a system for animals to adapt, which means there have to be beneficial mutations that are selected for, and they have to be very, very common for the types of diversity that we see today, even under a young Earth creationist lens, to evolve. Bergman's okay with that. Now the problem is, how do neurotransmitters evolve in the first place, and how do you get organ systems? That does not help you with the common ancestry of, like, all mammals, or like most animals, right? Now you've just shifted that problem backwards in time. And in my opinion, that acquiescence kills Bergman's young Earth creationism, because the same changes that were at play to get maned wolves and bush dogs from a generic canid ancestor could get you chimpanzees and humans from a generic Miocene ape ancestor. It is the exact same process. Humans and chimps have the same organ systems. They have all of the same bones. They have all of the same types of neurotransmitters, right? Like, you're just adapting on a common theme for new environments. So that's kind of game over. But Jerry doesn't care about that. He sort of pantomimes the where's the animal that has half a heart or half a stomach or half a liver argument by saying animals either have spleens or they don't. And then he talks about how there's no good explanation from the evolutionists as to where the 4-chambered or 3-chambered or 2-chambered or 1-chambered or quote-unquote 0-chambered heart comes from.
So I thought, well, gee, evolutionists probably have a good argument to show from 1 chamber to 4 chambers, and it turns out they themselves admit they didn't.
But that literature just is out there, and he does the same thing with the origin of the tongue, where he's like, we have no idea where the tongue came from or how it evolved.
And so from my research on the tongue, indeed, they admit we have no idea how the tongue could have evolved, when it evolved, and indeed the steps of the tongue evolution were totally baffled.
And it's like, dude, it's not my fault if you're not up to date on the literature on this. Fortunately, Chris is just like, I don't really care about talking about any of that. That's not the topic of the debate. I can come back and talk to you about the origin of any organ system you want at another juncture. We're talking about the origin of species for this debate, and he maneuvers into asking a question.
And so I want to talk about like speciation that can occur within kinds. Do you think that speciation can occur within kinds? Do you believe that there was speciation that occurred within kinds for the pairs of animals that came off of the ark?
Jerry kind of dodges this question by saying, well, it depends on how you define speciation, because what a species actually is very difficult to define. Now, as a species hater myself, I do concur.
Well, you have to define speciation.
But for the context of this debate, I think everybody knows what we're talking about here, right? It doesn't matter how you define species, whether you're using biological species concept, evolutionary species concept, the phylogenetic species concept, pick your favorite, use all of them. Can any of them happen? Can all of them happen, right? That's kind of what Chris is asking. But Chris agrees it is hard to define a species, and so he moves on and asks a different question and gets a similar answer.
What, like, for instance, how many kinds do you think were on the ark? Do you have any idea? And where do you put the line at kinds? Like,
I haven't looked at that.
Okay,
question.
so like the Cricetidae and the Muridae right here. So like the true hamsters, the voles, the lemmings, the muskrats, and the New World rats and mice. Answers in Genesis considers this to be a separate kind from the Muridae, which is the many species of mice, Old World mice, the Old World rats, and the gerbils. Like, do you consider these to be separate kinds?
I really haven't looked into that at all. Again, I just don't have. I know there's a lot of arguments on what group is part of this species, etc.
Chris takes some time and explains again the problem with created kinds and how at the family level, especially applied to rodents, it's extremely difficult. And then he asks Jerry again, like, how do you tell whether or not organisms are part of the same kind or not? And Jerry gives this sort of lukewarm, pseudo-biological species concept kind of answer.
Well, I would say if they can interbreed pretty regularly, then they're probably one kind. But if they can't, then they're not, less likely to be one kind. But again, I look at the flexibility. We see a lot of flexibility in the living world.
So no, he doesn't know. He's like, I could recognize it when I see it, but there isn't a distinct definition of a created kind. It's impossible to tell. He goes on this very odd sort of tangent about like the differences genomically between people who were brought over due to the transatlantic slave trade and people who are still living in Africa, to which Chris is like,
but they're all the same species. So we're talking about like how speciation occurs, and that was kind of my question, right? So if you consider like, so if because you know we're talking about like nearly 2,000, 3,000 different species just within these 6 families, right? At least 2,000 that have been described, and some of them can interbreed, but most of them cannot. You consider those to be separate kinds? So Noah had on the ark a male and a female of each of these kinds?
There are a lot of reasons, though, 2 animals can't interbreed, and they may not be able to interbreed for a lot of reasons which have nothing to do with their ability to do so.
He pivots again, I have to say. It reminds me a lot of the King Crocoduck versus David McQueen debate, which I'm sorry you have to hear this from me as well again, but David McQueen has since passed away. And that makes me really, really genuinely very sad because I really liked David McQueen. I thought he was a really fun, sweet-seeming guy. So, you know, in honor of David, at least one more time.
Tube.
But back to this, right? That doesn't help you though, Jerry, because that's just saying that reproductive isolation can actually evolve in kinds. So you can't ever tell what belongs to what kind using the ability to interbreed or not. Chris tries one more time to get a concrete answer, and he's like, is it biological species concept? Do you believe if they can't interbreed that they're different kinds? You know, the implication being that's going to be a whole lot of kinds, Jerry. And Jerry's like, maybe, sometimes.
But I hear you say that if they can no longer interbreed, that means they are separate kinds. Is that what you're saying?
Well, partially, but again, I must admit that creationists debate this among themselves a lot.
And then he goes into this next sort of tangent where he talks about how he just kind of doesn't really buy phylogenetic trees. And partially he doesn't buy them because of the concept of epigenetics, which Chris proceeds to try to explain to Jerry does not change the genome in that way, but that doesn't really break through. Specifically, Chris talks about those private alleles, right? These alleles that have clearly emerged since bush dogs and Maine wolves, again, we're back to bush dogs and Maine wolves, since they split from common ancestor. And Jerry's explanation for these private alleles is, I kid you not, a sort of soft pitching of like, maybe God designed it that way by like intervening, right?
My response is we don't really understand polymorphisms and why they are the way they are. We need to understand more in more detail polymorphisms, because there are so many. We say, well, this is polymorphism and has no function or no purpose. But I wonder if indeed some of these polymorphisms aren't a result in some way of design, in that indeed this allows the production of variety that we see in the natural world, which is, I would say, by and large, not random. There may be some randomness evolved, but I would say, again, we need to. And this, I guess, the difference between evolution and creation. Creation looked for design. They look for a purpose. They look for a reason. They look for a function. And scientists maybe should look at the creation world of view and say, well, maybe polymorphisms are more complex than we think. Maybe there's a reason for them.
God actively tinkering in the genomes of organisms, which is at least what I understand to be what Jerry is saying here. I guess I could be wrong. That's not science. You can't test it. It's not falsifiable. And so it is out of the question with regard to scientific investigation. So to summarize, in this conversation of the origin of species and evolutionary mechanisms, Jerry Bergman cannot define an alternative to species or a barrier or boundary to created kinds. He can't actually define what they are and how you tell one from another. He can't explain phylogenetic trees. He just kind of doesn't believe in them, and he doesn't believe in them when conventional scientists do it, and he doesn't really believe in them when creationists do them either. And with regard to explaining, like, the patterns that we see, he kind of just says, well, maybe God is, like, actively making it look that way. And in Chris Thompson's conclusion, he outlines, why that's inappropriate and why that's also quite frustrating.
This is not a scientific debate. Like, scientists don't debate the origin of private alleles. Like, we know that results from mutations that occur within the genome. Like, we're not going to try to pretend that there are angels that are tinkering with the genome. It just isn't the case. And that miraculous events to explain the origin of the Maine wolf in order and how it's different from the bush dog, just simply, that's religion. I mean, it is, and it's okay to teach that in church, but like, I don't think that that's appropriate for public schools.
Obviously, I skipped a lot of the repetitiveness of their back and forth in there. I think Chris did a wonderful job. I think he made his case superbly, and I think that the fact that at the end Jerry was appealing to, God being responsible in active tinkering for creating the evolutionary pattern, and that he can still be a young Earth creationist, God just did it that way, in a way that looks like evolution. I think Chris did kind of mop the floor with them. And you're about to get some deja vu, because in this next debate, we're going to watch Dan do the same thing.
We had this debate. The topic was common descent versus common design. I think it went really well.
If you're not subscribed to Dan, whose YouTube channel is Creationist, please do that. And also subscribe to Chris's YouTube channel. They're both phenomenal. But if you've been around here for a decent period of time, you probably know Dan already. He is an evolutionary biologist, and he's going to be debating Jerry Bergman on the topic of, what do the data support? Common descent or common design?
All right. Hi, everybody. So our debate topic tonight is common descent versus common design. And I'm here to tell you that we already know the answer to this question. It is common descent of all cellular life, not common design. And we're going to talk about exactly how and why we know that's the case.
I love Dan, dude. He cracks me up with this. He starts his debates or his sort of intros like this a lot, where he'll be like, the science is settled. The science has been settled for a very, very, very long time. But YouTube debates are fun, so that's why I'm here. Dan's thesis statement, the entire point of his opener, and the entire strategy that he utilizes is super, super simple. He's saying that common descent makes a bunch of different specific predictions as to what a phylogenetic tree or family tree at the species level should look like, whereas common design makes a very different series of predictions about phylogenetic trees or family trees at the species level. Because, under common descent, all species will nest within their given genera, which will nest within their given families, which will nest within their given orders, and everything can trace itself back to a single universal common ancestor, no problems. But under the creationist model, you have multiple different created kinds which are not at all actually related, that are being force-fit into a family tree. So what you should get, statistically speaking, is a whole lot of mess. Dan starts off with dogs and wolves, because both he and Jerry accept that dogs and wolves are actually related. So the phylogenetic tree that they should produce is a real tree. It is not an artifact.
So what we have when we're looking at all the dogs, and when I say dogs, I mean, dogs, wolves, maybe coyotes, whatever, wherever you want to draw the line, right? We agree they share an evolutionary history, so we have a shared pattern of variation within those common ancestors, right? Each node, each branch point represents a common ancestor between the descendant branches. So what we expect to see, what we predict when we construct our phylogenetic trees, is a hierarchical pattern of similarity with strong branch support, that is to say, strong statistical support, and very little polytomy, which is a branch point with more than two branches leaving it, where you have three or more, which just means you don't know what the relationship is. So, for example, if we look at a tree like this, right, maybe we've got dogs down here at this rightmost blue branch, and then we have wolves here, and they're more similar to each other than they are to, like, say, coyotes is maybe this kind of purplish indigo branch right here, because those dogs and wolves actually share more of their history together than either of them share in common with the coyote. So we expect this nested hierarchical pattern when things share common ancestry or within a created kind, right? And that's just how phylogenetics works, and we agree on that. We just disagree on where it stops operating.
We know what phylogenetic trees should look like, the pattern that they should throw when they're reflecting reality. So Dan's like, we agree with that. They look like this. It's all fine and dandy. If common descent is true, right, that pattern should reflect across all of life. And if creationism or separate creation is true, then we should get a very different signal.
The common design or separate creation position or hypothesis leads to a different set of predictions, because when you have two separate created kinds, they don't share an evolutionary history. There's no shared pattern or no shared history of variation, because all the variation since they got off the ark has developed independently in those two groups. Now, mutations are probabilistic rather than deterministic. We know that from direct observation. So the mutations that occur in those two unrelated groups will be uncorrelated with each other. So what happens if we were to try to do phylogenetics across created kinds? Well, you take your two groups that are unrelated, as I show right here, they don't connect, right? They don't share common ancestry, and you kind of mush them together. And what I'm trying to illustrate here is doing this, right? You have two trees and you basically smush them together. And because they don't actually share a common evolutionary history, you're going to get this weird pattern where you don't have strong branch support, right? You don't have high bootstrapping values, and you have lots of polytomy, right? It's just a big jumbled mess. There's no hierarchical pattern of similarity across the kinds.
This is phenomenal science, right? Because this sets us up for hypothesis testing. You have these two opposing predictions from the separate creation model and the common descent model, and you can then run these phylogenetic analyses and see what kind of patterns you get. And it will not come as a shock to you that we have run those phylogenetic analyses many, 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 many times.
We've made all these phylogenies. The question is, what does the data show? Well, here are the phylogenies. Two of them are within a kind. Two of them are across kinds. We should be able to tell which are which.
That's right. I took that part out of the beginning because I wanted you to see it here. But Dan is going to have Dr. Jerry Bergman tell us which of these phylogenies belong to separately created kinds that are erroneously shoved together, and in those cases they're both primate groups, which is especially relevant, and which of these phylogenies involve groups that Jerry and Dan would agree belong to the same created kind, which Dan says they're from the order Rodentia. This is a super creative way of arguing for common descent over common design, right? It's like posing the question to your opponent and saying, you should be able to tell the difference here, right? Tell me the difference between what is designed and what is the result of evolution. Because from an evolutionary perspective, there's no design involved, at least not mechanistically speaking. But from the common design perspective, some of the change that you see is the result of natural mechanisms, and some of it is designed in from the beginning. So how do you actually tell the difference?
This is just a summary, so let me just go right to the wrap-up. This is how science works. You construct a hypothesis, you make predictions, you test them. Here we have specific predictions. Phylogenies within a single kind will be distinguishable from phylogenies spanning two or more kinds according to a common design hypothesis. Is that the case? Let's see.
Jerry's opener is largely the same. There's a little bit of hominin stuff in there that I like, but I would rather cover that later in bulk, and he has a presentation just on hominins, so we'll save that for then. And then it goes to Dan's rebuttal. In his rebuttal, he gives Jerry an opening to tackle what his big questions are, right? Like, how do you tell the difference between common descent and common design when looking at phylogenetic trees, which are just family trees at the species level? And many times, especially more recently, they're done with the entirety of the genome, or at least the overwhelming majority of the genome, having been sequenced already. But Jerry does not care about that because he doesn't believe that phylogenetic trees actually work, and he doesn't believe that they work when people like Todd Wood do them, who is a young Earth creationist, and he doesn't believe that they work when evolutionary biologists or geneticists do them either.
Okay, thank you. It's interesting that creationists love these phylogenetic trees. Todd Woods has done dozens, many dozens of these. And I have to admit, I'm critical of both creationists and of evolutionists when they use these. I find there are major problems with them. And I pointed that out, not only the evolutionists, but also the creationists. And there are a number of problems. Number one, judgments are made relative to where people are, what genes you use to make the tree, what structures you use to make the tree, and so on. And you can select the structures you want, certain structures, and get a certain tree. You can select other structures and get another tree. And so I have to admit, when I look at these phylogenetic trees, I'm right away very skeptical, and I don't put a lot of weight into them.
I bet that he would accept that they work within humans, that you can make a human phylogenetic tree by looking at specific highly variable genes, but that's neither here nor there because, as I just said, many of these most modern phylogenetic trees are created having sequenced the entire genome, right? So you're not picking and choosing genes, you're using all of them. But Jerry spends a big chunk of his rebuttal talking about how the tongue can't evolve again.
By the way, hypothesis, I had a hypothesis not too long ago that indeed there should be evidence for the evolution of the tongue. And so what I found was very clear in the literature, there's simply no evidence at all that we moved from a tongueless creature, a fish. Fish don't need tongues, they open their mouths and forward, to a tongue. And of course, without a tongue, it'd be pretty hard for mammals, in fact, for many animals to survive. So therefore, indeed, the literature pointed out, supported my position, and therefore my hypothesis was shown to be correct.
Brother didn't look too hard. Again, like, you can hop on Google Scholar and you can be like, evolution of the tongue, and a million results pop up. You just have to read them. After Jerry's rebuttal, it finally goes into open discussion, and Dan can kind of put the screws to him a little bit. And what he does is really interesting and I think pretty smart. Dan goes step by step through what they have to agree upon in order to actually test this prediction. Like, can we agree that common descent and common design have these separate predictions? And can we agree that these are the predictions that they make? And can we agree that as a result, we can turn to these phylogenetic trees and see which idea they support?
What I want to first make sure is that we agree on the predictions that the two competing hypotheses make, right? So we've got, I say all cellular life shares common ancestry. You say within limited groups you have common ancestry, but each group is separately created or separately designed. So then the predictions, because the mutations are approximately random, then we should see uncorrelated mutations that occur in those different groups. Let's start there. Can we agree on that? That like in this lineage and that lineage, separately created, those mutations should happen independently and uncorrelated from each other. Can we agree on that?
That seems reasonable. Yeah.
Okay, cool.
You can really feel the "it's not my wallet" meme building up as tension grows.
So here we have our trees, and I will tell you that two of them are for rodents. One is, I think, one is mice and rats, and one I think also includes gerbils. And then two of them are, one of them is all simians, and another is all primates, which I think we agree that, I think you would agree in the creation model that all simians and all primates constitute more than a single created kind. It's two or more groups in there. Whereas like rats and mice, I hope I was okay in assuming that we would agree that's a single created kind. I hope that was an okay assumption.
The thing is, Dan, from the Thompson debate, we actually don't know. We do not know if Jerry accepts that rats and mice are related. We don't know if he thinks that they belong to the same created kind or not. I had always assumed that creationists are okay with them being the same created kind because, like, that tends to be the position that they hold, but we just don't know if Jerry is down with that or not.
The thing is, because we agree that mutations are probabilistic, and we agree on the consequences of that reality in terms of the structure of phylogenies when you're comparing across created kinds compared to within, with the separate ancestry model, the common design model, we should be able to tell which of these two trees is which. Can we do that?
The moment of truth, right? It's Dr. Bergman's opportunity to say which of these phylogenetic trees belong to the same kind and which ones are inappropriate mashups of kinds that are not actually related. And his answer is kind of hard to discern. I'm going to play it for you so you can hear it.
Yeah, A and B are similar. B and C are similar. And B and C look like you would say they don't have common ancestry. A and B do have common ancestry.
A and B have common ancestry. So I actually have to pull up my
No,
answer.
no, A and C, sorry. A and C have common ancestry. B and D do not.
Okay, so the point is they all share common ancestry. That's why none of them stick out like a sore thumb as an inappropriate phylogenetic tree. But Dan and Dr. Bergman go back and forth discussing the nature of these phylogenetic trees because the fact that one of the phylogenetic trees lacks a root at the top kind of threw Jerry off. But at last we arrive at sort of the conclusion.
Now, the thing is, according to your model, 2 of those common ancestors are real and 2 of them are not. And we just agreed we should be able to tell them apart, right?
My creationist friends could, but I guess I can't. Sorry.
Okay.
But my creationist friends probably are more attuned to this than they could.
Okay. Folks, that is my point. That is the whole point right there.
Right, so no creationist can actually tell the difference between phylogenetic trees that are the result of common descent and phylogenetic trees that are the result of 2 created kinds being inappropriately mashed together, because all of them are actually related through common descent. The whole thing is common descent. That's why they throw the same pattern every single time, whether you're looking at a family of people, a bunch of different populations of people, a bunch of different populations of apes, a bunch of different populations of primates, a bunch of populations of mammals, a bunch of populations of chordates. It doesn't matter. All of them are phylogenetic trees that rely on the simple assumption that genetics works the same way now as it did in the past, and all of them throw the same pattern, that organisms are all related through common descent, going back in time to a universal common ancestor. There is no gap. There is no point at which genetics seems to be throwing us a sign that says, nope, there's a barrier here. These 2 things are not actually related. And one thing that Dan does that's really cool is later he brings up in this debate that we actually have an example of what that would look like in viruses. Now, this isn't to say that viruses were separately created. It is to say that they have separate origins and are not related through common ancestry. So when you try to force them into a single phylogenetic tree, you get absolute noise in response. We do not see that pattern with all living things. Now, you might be wondering, why aren't we going over the rest of this debate? It doesn't seem like we went over very much of it. And the answer is that, and Dan covers this in his video, the entire second half of this debate is not really about, the debate topic. It's about a bunch of different other subjects that Dr. Bergman brought up in his intro, much like the Thompson debate, much like the Peasy-Myers debate. So I've been thinking about this with regard to my own debate with Dr. Jerry Bergman. It seems like a common theme in a lot of these discussions is that things get derailed and you end up talking about things that are not the debate topic. My debate topic with Jerry is extremely specific. It is, does the hominin fossil record support human evolution? Short, sweet, to the point. I imagine we're going to be talking a lot about fossils and a lot about gross morphology, and I think that because that title is so tight, it's going to be very difficult to talk about, mutations or creationist persecution in academia. So I got to thinking while I was consuming all this Jerry content, is there any place where he talks extensively about the fossil record so I know what his positions are going in? I want to know what he thinks, and I certainly hope he's going to do the same with me so we don't have to waste a lot of time explaining sort of our perspectives. We can get right into the data. And it turns out that yes, Jerry has talked a lot about his opinion about the hominin fossil record here online, and he talks about the fossils themselves. So let's do a little bit of that now. So Dr. Bergman has this presentation specifically on human evolution, and it concerns itself most primarily with Australopithecus and most specifically with Lucy. And I've watched a lot of creationist content covering Lucy, and so I really need you to hear me when I say that this is one of the most incorrect presentations that I have ever seen with regard to this hominin. I was listening to it at the gym, and a few times I had to stop and rewind it because I kind of could not believe what I was hearing. So let's get into that. This presentation is also on Donnie or Standing for Truth's channel, and as always with these kinds of things, Jerry starts it off with a Bible verse. He talks about Genesis one, and how God created man in his image, and how that's the basis. And so that's sort of the framework that he's working from. You know, human evolution cannot be true if you are taking a literal interpretation of Genesis as Jerry is. And then he goes on to this.
And ironically, there hasn't been that much consideration of this issue among creationists. And a good example is this book, Apes as Ancestors, which I recently completed with Peter Line and Jeff Tompkins and Daniel Biddle.
Oh boy, I can't think of any way that I would rather spend my time than read a book on paleoanthropology that has Jeffrey Tompkins on there as an author. I knew that Dr. Bergman had authored a book on like human evolution, sort of like a Contested Bones style book, but I had no idea Tompkins was on the book, and I had no idea Dan Biddle was on the book. Like, what does he know about hominins? For those of you who don't know, Jeffrey Tompkins is the guy that tried to propose for many, many years that like conventional science had it wrong and humans and chimpanzees were only like 85% similar to one another. And then it turns out he was wrong on all of that, and the entire creationist community has been slowly abandoning him and his number, thanks in part to me specifically.
And you'll notice that one looks like a male, the other looks like a female. Well, Lucy was the most famous by far example of human evolutionary link. And the problem with that is they don't know if it was a male or female. They don't have enough body parts, skeleton parts, to determine what sex it is.
Dr. Bergman goes off for a little bit on how if you accept human evolution, then it like decimates Christianity. We've talked about this a zillion times on this channel. No, it doesn't. There are plenty of Christians who accept human evolution and all of conventional science, and they make it work because they have different interpretations of scripture than people like Jerry does. And then he launches into talking about Lucy, which is the primary focus of this presentation. Now, Lucy is a member of the species Australopithecus afarensis. She is one of the most complete partial skeletons that we have from the genus, certainly the most complete from that species, and she is a wonderful, wonderful, wonderful piece of support for human evolution because she is a bipedal animal, a bipedal ape that has many primitive characteristics in combination with many modern characteristics. To put it very simply, she is an ape with a brain that is slightly larger than that of a chimpanzee, despite having a similar body size to a chimpanzee, but that walks completely bipedally when on the ground and has the entire suite of characteristics that denote bipedalism in like humans. We're going to get into that more later, but first Jerry wants to tell us why Lucy is sometimes represented as a male and sometimes represented as a female.
Problem with that is they don't know if it was a male or female. They don't have enough body parts, skeleton parts, to determine what sex it is.
Nope, the reason why Lucy was initially proposed to be a female is because she had typical characteristics of female apes, including more gracile facial features, a smaller overall body size, and feminized canine teeth. This assignment is pretty robustly supported because we found other members of Australopithecus afarensis, and they fall into two pretty neat categories. One is really big, and one is pretty little. This difference between males and females, this sexual dimorphism in body size, is pretty extreme. It's like close to the level that we see in gorillas, about intermediate between chimpanzees and gorillas. And yet, what makes Australopithecus afarensis pretty interesting with regard to its dimorphism is that, like humans, they're not dimorphic in their canine teeth. So you have an ape that is sexually dimorphic in body size, which is a more primitive feature of hominids, and is not sexually dimorphic in canine tooth size, which is derived for hominids. Almost like it's transitional in its state of sexual dimorphism. How weird. Now, I'm contractually obligated to let you know that I study sexual dimorphism in Miocene apes, so the group that immediately precedes the hominins, like, for example, Australopithecus as a genus, and I'm of the opinion that sexual dimorphism is, like, pretty much in flux all the time, depending on environmental factors and what that does to group size and competition. So I think that sexual dimorphism goes like this, so it's not necessarily a great characteristic to use as like a transitional feature. But one thing that isn't in flux is a reduction in canine teeth in both sexes that then sticks around for millions of years. And that's what we see happening in Australopithecus, where males and females both have very small canine teeth in comparison to other hominids. And that just seems to stick around and carry on through the rest of them as well. Jerry talks for a little bit about how when him, Jeffrey Tompkins, Peter Line, and Dan Biddle wrote their book, their goal was to survey the paleoanthropological literature, take claims that paleontology generally makes, or rather paleoanthropology generally makes, and then look at all of the opinions and then see which one they thought was the most correct. And their conclusion is everybody disagrees too much, so we kind of can't trust any of them. And I think that that's really silly for a couple of reasons, not the least of which that there are many things that all paleoanthropologists agree on. One of which being that Australopithecus, or Lucy's genus, was bipedal when it was on the ground. I wonder what Dr. Bergman is going to have to say about that. He goes on and talks about acceptance of evolution by denomination. We don't need to go over that. He talks about the Jeffrey Tompkins number, the idea that humans and chimps are only about 85% similar to one another when looking at the entire genome. That's just not true. I did the work for this myself. I compared billions of base pairs in order to come to that conclusion, and that conclusion has been scooped up by other young Earth creationists, like, for example, Rob Carter. And I challenge you to find anybody besides Jeffrey Tompkins' friends here online who are still touting that number, because it turns out Tompkins was just, really, 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 really wrong on it because his methods sucked really badly. Hilariously, when Dr. Bergman is bringing up the Tompkins number and saying that this National Geographic article that states humans and chimps are about 99% similar when looking at coding DNA base pairs, which is true and still is true even using Tompkins' methodology, he says this.
The difference actually is about 500 million base pair differences. In other words, there's an enormous difference between humans, human DNA, and chimp DNA. You wonder, why in the world do they state this something that's been wrong for decades? I don't know any reputable geneticist that would make this claim at all today.
Put that statement in your back pocket, why someone would say something that's been wrong for decades. I think we're going to use it later, but also any reputable... Dr. Bergman, all of them say this. There's a paper that was on the complete sequencing of ape genomes that came out last month, and that paper said this. It's the most up-to-date number with the most up-to-date genomes. You guys just gotta accept the fact that Tompkins really whiffed it on this one. Dr. Bergman goes on to talk a little bit about how Lucy's skeleton is actually super incomplete, and he makes this claim that she's only 20% complete.
Now, what did they actually find? Well, hopefully you can see the picture there. What's brown would be the bones they found, and the white would be what was added. So you can see the majority of the skeleton was added. In fact, you really don't even have a half of a side.
Okay, that 20% number is just wrong, but can we just appreciate that if you had half of a side, you would have the whole animal, because mammals are bilaterally symmetric. We're reflected down our midline. So if you had one side, like the whole right side, you know what the whole left side looks like. Now, to get to that 20%, right, the official reported number is that 40% of Lucy's skeleton was found, and that if you reflect that across the midline, about 80% of the skeleton is represented. So how does Bergman get it down to 20%? I'm actually not sure. I've watched this section a few times. I've tried to do the math on my own. I don't know if he's taking the volume of specific bones so that he can take fragments into account. It's pretty unclear to me. Ultimately, at present, it doesn't matter though, because we have found Australopithecus specimens that are over 95% complete in the case of Australopithecus africanus or Australopithecus prometheus, the specimen called STW 573. And the morphology, genus-wise, of this specimen matches that of Lucy's. And those aren't even the only two most complete skeletons that we have. We also have members of Australopithecus sediba, MH1 and MH2, the Dakika child, a member of Australopithecus afarensis, Katanumu man, a member of Australopithecus afarensis. I could go on and on and on. But they only ever talk about Lucy because that's where all the talking points come from. They can't deviate from that.
So of the 47, they found 47 bones. There's 207 in humans and 47 of 207. And of the 47, most were small fragments.
Yup, and do you know what fragments often do in paleontology? They fit together with other fragments, like pieces of a jigsaw puzzle. That's why you can have like 20 fragments and then also have the femur, because the fragments form the femur.
And so they have a lot of fragments. Therefore, there's a lot of guesswork that was required in order to determine what Lucy was like.
Imagine implying that something like this is guesswork. Again, they fit together like the pieces of a puzzle. What about this, right? If we find half of Lucy's pelvis, do you think we can reconstruct the other half of the pelvis and get a decent idea as to how she walked? Also, he didn't mention it, but you can see on that slide that he says Lucy's mandible or her jaw is more like a gorilla's. No, it isn't. We know this. It's obvious. So next, Bergman is going to talk about the Laetoli footprints. The Laetoli footprints are a trackway that are assigned to the species Australopithecus afarensis, and creationists think that that's dumb and silly because Lucy didn't have any hand or foot bones with her, so she could have been a knuckle walker, and so evolutionists are just forcing the Laetoli footprints to be assigned to Lucy because they need that to be true, and because these footprints look extremely human.
And you can see a huge difference between in this case, the feet of the apes and the feet of a human. Well, that's the footprints they found, and obviously that is the footprint of a human, not of an ape.
Humans are apes, first of all. Second of all, that's not the foot of a human, that's a foot of a biped. There is a huge difference. Third of all, we do have metatarsals and metacarpals that are associated with Lucy, and we have many, many, many more foot and hand bones associated with other members of Australopithecus. And fourth of all, we have investigated the Laetoli footprints. We've investigated the topology of the feet, and we've investigated the actual biomechanics of the track makers. And they're intermediate. The track makers' foot morphology and the way that they moved was like that of neither chimpanzees nor humans. It's more similar to humans than it is to chimps because the track maker is a biped. It's not a human, it's just something else that walks upright. You know, like genus Australopithecus, who has all of the characteristics of a biped when they're on the ground. I know you're wondering, why hasn't she done this spiel yet of all the characteristics that denote a biped? And it's because Jerry's gonna go through them. We're gonna go through them with him. In fact, the Laetoli footprints are Jerry's segue to talk about the morphology of Lucy, and I guess by proxy Australopithecus. He doesn't mention any other Australopithecus specimens, but I think that's kind of the idea. In fact, at one point he's like, Lucy, the individual specimen, was too small to have made the Laetoli footprints, so therefore it couldn't have been a member of Australopithecus. Need I remind you that Lucy is a female, and a very small female at that. Not all Australopiths are female, the males are quite a bit bigger, and there's also a lot of variation within males and females as sexes within Australopithecus. It's kind of like how you can have really tall women or really short women, and also really tall men and really short men. So Jerry wants to walk through characteristics of Lucy. He starts with the skull, right? So he begins with the cranium and the mandible, and his conclusion is basically most of it's made up because it's too fragmentary to really say anything, but the fragments also simultaneously, while not being able to tell us very much information, tell us that she was definitely just an ape.
Skull you can see in this picture here is a few small fragments of bone, and you see a large amount is filled in by the investigator. And these fragments here, judging by the fragments that are there, and if we look especially at the temple area, they indicate they are from not an ape-like person, but an ape, period. Which is what I conclude Lucy was, simply another ape.
Yes, Lucy was an ape, so are humans, but what he means when he's saying this is that she is an ape and humans are not apes and therefore they're not related. That's what he's meaning when he says this. This talking point by creationists really irks me, this idea that researchers just made up the rest of the skull, filled in the blanks with what they needed when they discovered Lucy's comparatively very fragmentary skull as compared to the much better represented postcrania. But, like, that's not what happened, right? We had, at the time of Lucy's discovery, several well-preserved crania, and the most famous one is Mrs. Ples, which is a member of Australopithecus africanus. Looking at the teeth of Australopithecus africanus and the teeth preserved in Lucy told the researchers that these were members of the same genus, and so they were able to compare the shared aspects between the two and base the rest of Lucy's skull off of Australopithecus africanus, her very close relative. But also it doesn't even matter because we found more complete crania of Australopithecus afarensis, or Lucy species 2. Jerry moves on to talk about how Lucy should kind of accurately be represented, and he shows the recreations of Australopithecus afarensis from the Creation Museum in Kentucky, the one that I have spent a lot of time on this channel explaining why it is inaccurate.
And here again, we see the Lucy walking from what we conclude, given she was an ape. And the other slide, you can see the green. The green are actually the body parts that we found. And so you can see we don't really have an adequate number of body parts to determine that indeed she walked regularly on 2 feet.
To be abundantly clear, you cannot reconstruct Lucy or any of the Australopiths as knuckle walkers. You just can't do it. The location of their foramen magnum, the bowl shape of their pelvis, the presence of a valgus knee, the 3 arches in the foot, the inlined big toe, the limb proportions, the S shape of the spine, the countless muscle attachment sites that are found in humans and not in other living apes, like the anterior inferior iliac spine and the linea aspera, among others, tell us that Lucy was a biped when she was on the ground. That does not mean that she did not climb, but it means that when she came down from the trees, her entire species, her entire genus, were bipedal. This is supported not just by Lucy the specimen, but by the other partial skeletons and many complete skulls that we have from genus Australopithecus. I'm at the point with this where I believe that it is just plain deceitful for Answers in Genesis and the Creation Museum to have Lucy's species represented as they do. They should know better. I believe that they have come across the research that shows them that they are incorrect on this many, many, many times, and yet they proceed onward with a completely indefensible recreation. I think that counts as lying personally.
Anyway, you can see now the skeletons of an ape and a human. You can see how enormously different they are. And therefore, you are looking for these differences when you find skeletons of supposed ape men.
Well, yes, that is a human skull and a chimpanzee's skull, and chimps and humans are not the same thing, so they are different and we expect differences. It is not accurate to say that we expect to see a chimpanzee condition in the past and then a human condition as we move forward towards the present, because chimpanzees have been evolving for just as long as humans. Instead, we should see a generic Miocene ape condition derive through time into modern chimpanzees and modern humans, which is exactly what we see.
And of course, the problem is, in most cases, all we find is fragments, and therefore there's a lot of speculation and guesswork that fills in the details.
I intend to ask Jerry at some point in my debate with him, what guesswork is involved in this? What are we guessing at with this?
And here we can see primates from my left, in the middle, Australopithecus, and then Homo sapiens. So you can see from the skulls, they are very different.
Ooh, this is starting to make me irritable. From left to right, you have a modern chimpanzee, which is a type of primate, Australopithecus, which is a type of primate, and Homo sapiens, which is a type of primate. And yes, they are different because they belong to different genera, but they are all primates, they are all apes, and 2 of the 3 are hominins. How do we know that, you might be thinking? Well, the answer is super simple. Because when you compare those 3 organisms, and we have all of the bones represented for chimpanzees, for genus Australopithecus, and for humans. To be clear, we have every single bone represented in some form, in some specimen for Australopithecus. And when you compare all of the bones, the animal Australopithecus shares more with humans than it does with chimpanzees. That's why they're both hominins and chimps are not. Okay, you guys, so this next part is pretty unbelievable to me, so I'm going to give you a little preface, and then we're going to watch it, and then I'm going to react to it and explain to you what the problem is. So Jerry starts talking about how you can tell how an animal moves by its skeleton, which is totally true. And with regard to the skull, there is one feature that is extremely helpful, and it's the hole at the base of the skull, which is called the foramen magnum. This is where the spinal cord exits the skull, and in animals that are quadrupedal, it's located way here posteriorly at the back because they're they're quadrupedal. They hold their heads relatively level with the ground. In chimpanzees, it's slightly more forward because chimpanzees knuckle walk. The way that their spinal cord comes out is sort of at an angle, just like that, and that's because their back is at an angle because their arms are longer than their legs are. Humans, though, walk upright, which means that our head has to sit at the very tippy top of our vertebral column so we can survey the landscape around us and hold our body in a singular column, and because we're only using our legs instead of our arms, we're bipeds, so we have a very anterior foramen magnum. So when you level the palates of a human and a chimpanzee, what you can see is that the chimpanzee's foramen magnum is way more posterior than what you see in a human. In this sort of inferior view, it's super duper obvious. So, what that means is that we can look at the location of the foramen magnum in hominins, like Lucy's genus Australopithecus, and deduce how it held its body and thus how it likely moved around. So, let's hear Jerry for a minute.
And when we look at the bottom of each of the skulls, we can see now the placement of the foramen magnum in all four examples compared to humans. And this dynamically shows the difference between this. So when you're looking at skulls, a key thing to look at is the location of this foramen magnum, which they did on Lucy, and of course from what evidence we have, which is not much because there's not many skull pieces we use, indicates she was an ape.
Excuse me? That's not even true for the most primitive hominins, Jerry. I'm getting real sick of having to pull the skulls out. No, I'm not. Those hominins in order are Sahelanthropus chilensis, Ardipithecus ramidus, Australopithecus africanus, Australopithecus afarensis, Paranthropus boisei, followed by Homo habilis and Homo rudolfensis. All things that creationists variably charge as being just apes, all with anterior foramen magnums compared to chimpanzees. Now, I know what you might be thinking, but Erica, Jerry's just talking about Lucy, for which the foramen magnum is not preserved at all, so I don't even know why it's being brought up. And to that I say, nope, he's looked at all of them. He says so earlier in the video.
And this you can see is relatives of Lucy. And for our study, we looked at all of these different relatives and found the same problems with all of them. But for now, we're focusing on Lucy.
That problems he's referring to is that they're fragmentary and also ape-like, which, no, that's not true for either.
A major difference, too, between apes and humans is the brain size. When we look at Lucy's brain size, from what we can determine, it was close to a chimpanzee.
In preparation for this debate, I played around in R a lot. That's a statistical program that also helps you make some really lovely little graphics, and I wanted to play around with plotting humans against other living hominoids, and then also brain case estimates through hominin evolution, just to see what that would look like. First, I compared humans to other living hominoids. Specifically, I looked at gorillas, so genus Gorilla, genus Pongo, genus Pan, genus Hylobates, and genus Symphalangus, and I got this lovely chart. You can see the means are marked very clearly, and humans have a much larger average brain case size than the rest of the hominoids, like about 800 cc's higher. Humans average between 12 and 1400 cc's, whereas chimpanzees are averaging between 300 and 400 cc's. This holds even when you take body size into account, so I used the standard equation to calculate the encephalization quotient for mammals and applied it to apes, and as you can see, humans are still significantly higher than the other apes. Then I decided to plot all hominin brain case size estimates against time to see if we see an increase over time, with humans marked in black in the present and chimpanzees marked in black in the present as well. And what you can see, I hope, is pretty obvious. There is a stark increase through hominin evolution in brain case size. It's a little bit of an increase throughout Australopithecus, but it really gets going in early genus Homo into later Homo erectus. Critically, there are members of each genus which overlap with the genus before it. So the highest Australopiths and the lowest members of genus Homo overlap. A creationist like Jerry Bergman or perhaps Donnie of Standing for Truth might say, well, then just assign those lowest members of genus Homo to Australopithecus. That just moves the problem up, because then still the lowest members of genus Homo, the new lowest members, overlap with the highest members of what is now Australopithecus, and so on and so forth. It's a real issue. And I also ran the same analysis with body size adjusted brain case sizes, or the encephalization quotients, as I did with living apes. And wouldn't you know it, Australopithecus and Paranthropus, or the Australopiths generally, have higher encephalization quotients than any living non-human ape does. This makes sense behaviorally because Australopithecus is the genus that we see the first stone tool use with. So it makes sense that they would have larger brains for their body sizes than chimpanzees, because the types of stone tools associated with Australopithecus are more advanced than the types of stone tools that we see associated with chimps today. So it is a massive oversimplification, and is also, I would argue, grossly incorrect to insinuate that Australopithecus afarensis, or Lucy's species, had a brain that was just like that of a chimp.
Other differences are the internal ear. Since humans walk upright, the ear has to be designed differently so that they can walk upright without getting dizzy, and apes likewise, so they can walk on all fours without getting dizzy. And when we look at the evidence we have, which is scanty, it indicates that Lucy was a chimp.
I know you already got jump scared by the classic creationist trope that other apes don't have white sclera, which they do. It's a part of their natural variability, but we'll get there here in a minute. I want to talk about the semicircular canals first. Yes, Lucy's genus, Australopithecus, has semicircular canals that reflect something that is still spending some time in the trees. In fact, we see aspects of that in other parts of their morphology as well, such as their curved phalanges or more dorsally oriented scapula, right? That's because if you are a small-brained ape living on the savanna, when you come to the ground, you might be walking around bipedally, but you can't sleep on the ground. You still need to maintain some arboreal aspects, because that's where you're going to be foraging, that's where you're going to be sleeping and escaping from predators. If climbing is still that large a part of your life, you need to have semicircular canals that reflect that. Like, the consensus on Australopithecus as a genus is that this is an animal that spent most of its time walking bipedally on the ground, but still went up into the trees to sleep and forage and escape predators. This is the anatomy of something that walks bipedally on the ground but is still a capable climber. It is not the anatomy of a chimp.
Almost all, there's one minor exception, but almost all apes, you cannot see the whites of the eyes.
Nope, it's surprisingly common in other apes to see white sclera, and in one specific study of chimpanzees at Ngogo, around 16%, or 1 in 6 chimpanzees, had white sclera. I'm sick of this one, guys. Stop saying it. I even made a video about it, and they're still saying it.
And there we can see the skeletal designs for the hip area, the iliad, the sacrum, and so on, for a walker on 4 legs is very different than a human, which walks on 2 legs.
Yup, that's true. Sagittally oriented ilia and a general bowl shape for the pelvis with lumbar wedging in the back is generally characteristic of a biped, so why do all the Australopiths have them if they're not bipedal? Not to mention, all the Australopiths have that anterior foramen magnum, they have valgus knees, they have inline big toes, they have 3 arches in the foot, they exhibit lumbar lordosis, they have those muscle attachment sites that I mentioned earlier that are associated with bipedalism and exist only in humans to the exclusion of non-human apes. Like, Australopithecus as a genus has every physical characteristic that you need to be a biped when you are on the ground, and none of the characteristics that you need to be a quadruped on the ground. Full stop. I just don't know how you could possibly make the argument that it's some kind of quadrupedal knuckle walker.
And there you can see actually the chimp walking around, and you can see then the bones of Lucy are placed to some degree to give you an illustration of, indeed, the long hip with flat iliac blades is what they find evidence for in Lucy.
Oh, okay, that's how. Just by not looking at the fossils, not showing the fossils, just showing a picture of a chimpanzee and then saying Lucy's bones look like that. Trust me.
And there you can see for a human the curved iliac, and it's curved in such a way to where the gluteus medius, the muscle, can attach to the way so it can walk upright comfortably.
You mean like this? You mean like this, Jerry? The fossil that I'm showing on the screen right now, right? Australopithecus afarensis lucy, specifically is pelvis, which has a really short ilium and a really short ischium that looks more like humans than it does like chimpanzees, and like it's not close. Like that. So, Jerry goes on to talk about knuckle walking in chimpanzees and gorillas, which, if you're interested in this kind of thing, chimps and gorillas actually knuckle walk in distinct ways. And then he goes on to compare the metacarpals of Lucy versus those of chimpanzees, and specifically he makes a comparison of a human metacarpal to Australopithecus afarensis metacarpals, and he says this.
And one good example is the top example here is the finger bone they found from Lucy. It is curved. A human finger bone is not curved.
Yes, that's true. Lucy has curved phalanges because she still climbed in the trees. This is the case for, like, all members of Australopithecus and even some members of genus Homo. But what does that have to do with knuckle walking? Because knuckle walking is generally associated with the carpals and, at the very best, like the proximal metacarpals. Curved phalanges tell you that something did a lot of climbing in the trees. It doesn't mean that something couldn't be bipedal or wasn't bipedal. So if you find curved phalanges in the entire suite of morphology that denotes a biped, that tells you that you had a biped that still climbed in the trees. None of that has anything to say about knuckle walking except that the animal couldn't knuckle walk, because actually bipedal morphologies are in direct contrast with knuckle walking morphologies. They're not in direct contrast with climbing morphologies.
So therefore, this is one more evidence that indeed the skeleton of Lucy was of a chimp.
Nope, for the reasons that I just stated. Okay, we're going to finish it off with Bergman talking about one of the metatarsals, so one of the bones of sort of the sole of the foot, and he's going to show us a comparison of a human fourth metatarsal, Australopithecus afarensis' fourth metatarsal, a gorilla fourth metatarsal, and a chimpanzee fourth metatarsal. And this is important because the proximal portion of the fourth metatarsal can tell you a little bit about, like, how the foot is shaped, the arching of the foot, and how it probably was used in locomotion.
And there we can see Homo sapiens, the fourth metatarsal in this case, from Lucy, which is AL 333-160. And then we can see two other examples. So we can see the metatarsal from Lucy is much more similar to the other apes that we have shown here.
Okay, so I think it just had to have been a long day, because that slide, like Jerry's own slide, is depicting the fourth metatarsal being more similar to humans than it is to chimpanzees or gorillas, right? His slide even says 1.7 million years old, yet fully human. But verbally, he tells us that Lucy's fourth metatarsal is actually more similar to other non-human apes. So what does he think about it? I don't know. Now, you can visually see, for the record, that fourth metatarsal is more similar to humans than it is to other apes, like gorillas and chimpanzees. So Australopithecus afarensis' fourth metatarsal looks more like ours than it does to chimps or gorillas, but let's look at it from like a numeric sense as well. Ward et al. 2012 shows this in breathtaking detail. It doesn't matter which portion of the bone you're looking at, it looks more like humans than it does like chimps or gorillas, but not exactly like a human. It's not fully human, Jerry. It's transitional. There is a lot more in this video with Dr. Jerry Bergman, where he's giving this presentation. Well, not when he's giving the presentation. He finishes the presentation and then takes questions from the audience. There's a lot we could dig into there, but I don't have it in me. Right? I don't have it in me to go through this entire thing. Plus, we've been here for quite some time and I have a debate to prepare for. I feel pretty prepared for this debate with Dr. Jerry Bergman that I'm having, according to my calendar now, tomorrow evening. Pretty late. It's going to be here on this channel, simulcasted, so please come watch it if that's something that you're interested in. I'll be taking super chat questions from the audience, and I'll probably stick around for a little while afterwards to answer any more questions that you might have. It's going to be very, 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 very fun, I think. If you like what I do here on this channel, and if you've enjoyed this sort of deconstruction of a lot of Jerry Bergman's material and opinions, please consider supporting this channel in the free way, which is by liking, commenting, and subscribing. It helps the algorithm, believe it or not, suggest my videos to people who might find them interesting. If you want to help me in a more financial way, you can support me via Patreon. Patrons when I am on top of things, which I am not and haven't been for like 6 months now, get early access to videos usually, but they always get their names at the end of every video. And so from the bottom of my gentle and of course very modern heart, I'd like to thank you for being such a curious primate, and I'll see you tomorrow.